Today we're doing Khan Academy's MCAT Prep Cells Section Cytoskeleton Questions. Okay, the first question. A couple is told by their doctor that the reason why they cannot have children is that the sperm of the male lack motility because it does not have the structures responsible for propulsion. Which cellular structures are likely to be the underlying cause of this lack of motility? Microtubules, vacuoles, endoplasmic reticulum, or Golgi apparatus? So what do we know about motility of cells via a flagellar type thing?
So I think listening to a couple of other review things throughout the week that I've learned that for prokaryotes, there's like a flagellin protein that kind of does this for prokaryotes, but for eukaryotes, they want us to know that microtubules are I wanted to say innervates, but that's like totally a different definition. But they're the things that allow the motility of sperm. And while we're on this subject, do we want to talk about the relative lack of contribution that the sperm give in creating gametes?
No, we'll just it's a lot. It's a lot.
Like way less genetic information or something like that?
Yeah, it's like way more genetic information or something like that.
Way more. Okay.
So the answer here should be microtubules, and it is. Okay. Next question. Malformation of which cytoskeletal structures may be linked to an inability to contract muscle? Microtubules, centrioles, intermediate filaments, or microfilaments? Okay, so I remember that I think it's either intermediate filaments or microfilaments are in the sarcomere, which is a motor functional unit. Mm-hmm. Actin is in there, right?
Yep.
We've gone over this before. Actin.
And what is the thing that, based on sliding filament mechanism method or whatever, the other protein involved in that?
It's like myosin or tropomyosin or something.
Myosin, yeah. Actin and myosin slide past each other.
And I remember that.
And what do we say about microfilm?
I was just about to say film, actin, actors.
Yep.
Okay, so it would be microfilaments here. Yep. I also remember we had a great thing about intermediate filaments. We said that it's kind of like when you're like, if you're constructing like a concrete slab or something, you need to put in like the wooden structures as the support first, and it's like the intermediate support.
Yep.
Yes, so microfilaments was the correct term. Okay, so question number 3. The ciliary action of the cells lining the lumen of fallopian tube are responsible for transporting an egg from the ovaries to the uterus. Which cytoskeleton structures are linked to this movement? Microfilaments, intermediate filaments, microtubules, or flagella? So flagella is made of microtubules.
Yep.
Intermediate filaments, our understanding is structure.
Mm-hmm.
And microfilaments, if you have actin and I think myosin is a intermediate filament, or sorry, microfilament. I just don't think that those would be involved in it. It seems like it should be microtubules. Do you have any thoughts on this?
Yeah, I think microtubules are the things that are inside of, for eukaryotes at least, inside of both cilia and flagella.
Okay. Yeah, that's right. Cilia and flagella. We're, in fact, going to listen to a presentation done on cilia on Friday, and I'm sure they'll be talking about microtubules there. Very cool. All right, next question. The cytoskeleton of a cell can generate movements such as in cytokinesis or propulsive forces outside the cell, like flagella. Which structures are linked with these two movements? So the two movements are cytokinesis or propulsive forces. Is it microtubules and intermediate filaments, respectively, microfilaments and intermediate filaments, respectively, microfilaments and microtubules, respectively, or intermediate filaments and microtubules, respectively? So we know that like propulsion in a flagella, that's microtubules.
Mm-hmm.
So it's got to be either C or D. So the key is cytokinesis. What does that make us think? Like an intracellular movement. Is that going to be microfilament or intermediate filament?
I'm almost tempted to say microfilaments, like elongating of the cell during mitosis or something like that.
Actin spindles? That doesn't sound right.
Filament.
Film.
So that would be oh yeah, you're right.
Well, so intermediate filaments structure. Microfilaments, we at least know that in actin and myosin, they are involved in moving something.
Right. Cells against each other in muscles.
Right. I think I'm going to go with microfilaments.
Okay, I know microtubules has to be the second one.
Yes.
I'm going to go intermediate filaments just because I know that they are inside doing something.
Okay, 1, 2, 3.
Oops.
Okay, I got it right. So it is microfilaments are involved in intracellular movement, such as cytokinesis, but let's look at those hints to see if our thinking is correct. Intermediate filaments do not generate movement. They're structural proteins.
Okay. They are just inert.
Microfilaments are linked with cytokinesis. Microtubules form flagella. So I think that the microfilaments might be involved in, if you ever watch like an amoeba chase like a little bacteria or something.
Yeah.
I think that's maybe microfilaments that like form that proboscis. Oh, I remember that word. And then once it moves and if it wants to stay still, that's when you would start putting intermediate filaments out.
Very cool.
To lock yourself there. Okay, next question. What is the name of the phenomenon in which a white blood cell engulfs a pathogen? Which cytoskeletal structures are involved in this process? Kind of funny, we were almost just talking about that. Is it phagocytosis and microtubules, pinocytosis and microtubules, exocytosis and microfilaments, or phagocytosis and microfilaments? So it's definitely phagocytosis.
Mm-hmm.
Pinocytosis is like liquid, like drinking water, basically.
How do you remember that?
Pinot Grigio.
And
Drinking and
pina colada.
Pina coladas. Exocytosis means out. So putting something out, so it's the opposite of what we're doing.
Yep.
So phagocytosis, white blood cell engulfing a pathogen. Which cytoskeletal structures are involved in this process? Well, we kind of just, my guess in the last one was that it was actin and myosin that form the proboscis.
Mm-hmm.
So if that were the case...
Maybe it goes out and in.
Yeah.
All right.
So
I like that.
It should be phagocytosis and microfilaments?
Wow.
Okay, so that was correct. So let's look at the hints to see if our thinking was correct. Exocytosis is the releasing of substances. We knew that. Pinocytosis is fluids. We knew that. Microfilaments are used for cell movement. Okay. Phagocytosis is the engulfing movement that a cell will do to destroy a pathogen. Microfilaments are responsible for generating the momentum necessary for this process.
Okay.
So maybe that really is how it works. Okay, next question. Your trachea have short hair-like cell process. Your trachea have short hair-like cell process that move the mucus. Calling you out, Sal. Okay, what is the name of these hair-like structures and which cytoskeletal elements form them? Flagella and microfilaments, villi and intermediate filaments, microvilli and microtubules, or cilia and microtubules. So what is the name of these structures? I feel like it's either gotta be microvilli or cilia.
Yeah, we know it's microtubules, and I would say that because we know that the cilia are composed of microtubules, so I would have gone with that one.
Okay, yeah. So I'm not entirely sure what villi... Aren't villi the folds? It's not the things on the outside, but it's like your intestine has lots of folds.
So the hints say microvilli are the foldings of the cell membrane.
Okay, cool.
That increase the surface area of the cell, but are not responsible for movement.
Okay, so villi are macroscopic, meaning like if you looked at the intestine, you would see the folds.
Yeah.
But then within those folds, you have microvilli that are like, it's like a fractal. Like more folds. Okay. So, yes, so it's cilia, and we know that cilia, flagellum, those kind of things that have like any kind of movement, any kind of whipping motion, microtubules. And we got it right. Okay, next question. Aneuploidogens. I almost had that one. Aneuploidogens are chemicals that may disrupt gamete formation by interfering with spindle formation and the last phase of cell division. As a result, the embryo may have abnormal ploidy. Based on this information, which cytoskeletal structures and which cell division processes are affected by aneuploidogens? Is it macrofilaments and telophase, respectively, intermediate filaments and mitosis, respectively, microfilaments and spermatogenesis, respectively, or microtubules and cytokinesis, respectively? Okay, you're interfering with spindle formation in the last phase of cell division, abnormal ploidy. Which cytoskeletal structures and which cell division process is going to be affected by that? So spindle formation, that, it's actin, right? Or is it microtubules?
I think it's microtubules. I think it starts from centrioles, and then the microtubules come out, and that's what helps us elongate and eventually split into two. It's really cool to actually see the spindle form in like the confocal microscope, because it really just looks like a star. It's really cool.
Okay, so, well, that kind of narrows down. There's only one answer that has microtubules is the first one. But then, so cytokinesis. So, yeah, that makes sense. So abnormal ploidy, I think they were trying to kind of throw us off with the whole ploidy thing.
Yeah.
What they're really asking is, if you can't form spindles, can't form the microtubules, then you're not going to be able to, if you can't form the spindles, then you're not going to form microtubules. And that's not going to allow cytokinesis to happen.
Yep, exactly.
Okay. And we got it right. It was microtubules and cytokinesis. Okay, next question. Mutations can change the overall structure of proteins, predict the outcome of a harmful mutation that modifies the microfilaments of the cytoskeleton of a white blood cell. It would ameliorate the structural support of the cell. It would decrease its ability to phagocytose other cells. It would impair the spindle formation during mitosis, or it would result in decreased levels of centriole activity. Modifies microfilaments. Yeah, this is the same thing we keep talking about.
Yep.
The white blood cell chasing a little bacterium or something. So it would decrease its ability to phagocytose other cells.
I would agree.
And we got it right. All right, awesome. Next question. Which cellular function may be disrupted because of a malformation of the intermediate filaments? Resilience to compression forces, formation of the spindle, transportation of chemicals, or ability to divide? So we always talk about intermediate filaments as structure.
Yep.
That would mean resilience to compression forces. That one seems pretty straightforward.
Yep. The others seems like they would go with other things that we have talked about already.
And we are right. Resilience to compression forces. Okay, last question. Which cytoskeletal structure is linked with the movements of a macrophage? Glycocalyx, microtubules, microfilaments, cilia, and flagella. This one could be a trick question.
Yeah.
They could be trying to get you to say cilia and flagella, I think.
Right.
It's microfilaments.
I would say microtubules, right? Don't they? Isn't that what goes through cilia and flagella?
That is what goes through cilia and flagella.
Right.
So in a way there, isn't that like because they said both, like they're like microtubules would be the same thing as cilia and flagella, therefore you can't pick either? I
don't know. I wouldn't say that microfilaments would be responsible.
Well, so if a macrophage is a macrophage is a white blood cell,
right, that eats something else. Are you thinking about the eating process?
Yeah. Ooh.
But it's not asking about that, right? Let me make sure. Nope. I'm
going to say microfilaments. Okay.
Please explain.
It is microfilaments. So I think that whole motion process, since they don't have flagella, the way that they move is by like pushing something out and then pulling everything with it. And then, following the chemical gradient probably left by that bacterium. The hints say Glycocalyx does not generate movement and it's not present in macrophages.
I think that's just the outer sugared surface of the cell.
Yeah. Cilia and flagella generate movement, but not in a macrophage. That would have been microtubules. Okay, next hint is microtubules make cilia and flagella. And then finally, microfilaments are responsible for amoeboid movement of macrophages. Okay, so the amoeba thing was real.
Yeah.
It's that must have been where they first saw it, and they said that's called amoeboid if it like shoots out a projection and then pulls everything in that direction.
You guys ought to see Tall right now.
I'm very animated.
So let's summarize, because I want to have this in my noggin too.
Okay.
So for microtubules, what are a couple things we want to take away about microtubules?
One thing people often forget, centrioles and
Right.
pulling apart sister chromatids.
Yep.
Also, they're involved in flagella and cilia.
For eukaryotes.
Yes.
And they form those centrioles, like that you just said.
And I can't think of anything else that they do.
Yes.
I don't know where else they're present in the cell.
I think that's a good start,
Yeah,
at least.
definitely a good start.
So then microfilaments, we know that for our way of remembering it as film, includes actors, actin, and myosin.
It works.
And so that would also be, so instead of movement with the microtubules like the cilia and flagella, for microfilaments, it's an amoeboid movement.
Yes.
And that also is involved in taking in things in vesicles and letting out things in vesicles.
Yeah. So I'm starting to think we don't see, at least with eukaryotes, we don't see like a flagellar movement on anything that's trying to like eat something else.
Mm-hmm.
And you do see amoeboid movement, and that kind of seems like it's an efficiency thing.
Okay.
Because the way that, like we learned about runs and turns
Yep.
for bacteria, it's kind of like random, but eventually they'll get there.
Right.
But with like the macrophages and the white blood cells, you need to be more precise. So you need that amoeboid movement to move, which we just have to kind of remember. That means it's something inside the cell that's like pushing the membrane out. So that's actin and myosin.
So if we are the stage, if we are a theater, then they're actors in our play.
Ooh, there are actors in our play moving us from the inside. And then the final is intermediate filaments.
Yes.
Which you had a phenomenal... this is how I remember it now.
Okay.
That it's like when you're putting a foundation of a house.
Yeah.
Like if you want that strong foundation, you have to put up some kind of support system around the actual structure.
Yep.
Like the wooden thing around the slab.
Yeah, or like the pillows before you add the blankets and a pillow fort.
Absolutely. I mean, you're right. You're completely right.
Thank you. I like hearing that phrase.
Strong work, gang.